Oh,
Uh,
hi everyone. I'm Lipeng. I'm the today the host of Let's Discuss paper after reading it. I'm the first year's PhD student in Suchiya's lab, Mike.
Hi, I'm a second year PhD student in the same lab in Monash University, Chuyin.
I'm a third year PhD student in the same lab at Anson.
And I'm first year student, PhD student in the same lab as well. Yes.
Okay, so today I'm going to talk about a paper. The title is A Behavioral Paradigm for Measuring Perceptual Distance in Mice by Nakayama et al. So simply speaking, this paper introduced a behavior paradigms for mice to indicate the perceptual similarity between stimuli. In this paper, they use other stimuli. And they begin by such kind of impractical finding or argument like perceptual, which is perceptual similarity or perceptually similar object should elicit by similar pattern of neural activity in brain. So to understand the neural coding mechanism, the relative relationship or perceptual space is important to understand the uh, or finding the correspondence of neural activity in brain. So this kind of method is easy in human, but in non-human animal, it is very difficult. So it call for a more reliable paradigms to do this kind of perceptual distance measurement. And yeah, in this paper with such a paradigm, researchers are able to test both the domain and link them together. And I shall start with the actually the aim of the paradigms. Okay, the aim is given a pair of stimuli and see whether a mouse perceive it as similar. And how can we know that? Traditionally, we use kind of let me see, cross habituation paradigms. And the idea of this paradigm is they present a stimuli at first, then the mouse will explore it freely because because of their curiosity. And then they repeat this many times with a stand simile until the mouse lose the interest to the simile. Then they present another simile, which is the new one. If the mouse explore the time for the new stimuli increase significantly, that means that the mouse perceive this new stimuli as a novel and can discriminate it from the previous one. The longer the time, the higher the novelty or the difference between two stimuli. But the problem is the habituation task take time. The process of habituation takes a really long time. And if we want to compare the relationship or the similarity between multiple stimuli, some stimuli must present more than once, right? If we want to exhaust all combination. So we need many mouse. Otherwise, for only one mouse, we can only, oh no, many mice. Oh. Otherwise, for one mouse, we can only test a very small subset of stimuli. So in this study, they take another way. They introduce a paradigms called delay match to sample behavior panda, which retrieved from cognitive domain, which is originally as a cognitive paradigms, where a mouse is presented with a stand simile. Then after a delay period of time, um, um, when two or more comparison or comparable stimuli are present, the mouse is rewarded for choosing the comparison that match the previous one, the sample. Okay. In this study, specific in this study, uh, mice are trained in this kind of way. If they found two sequential presented stimuli are the same, they will lick the center water port. if two things are different, they should not leak, which is no go. Then after they do this kind of no-go behavior, they show this kind of no-go behavior, and it is correct, which means that two stimuli are literally different. They are rewarded by the water. Okay. Then the probability of leaking will transform to distance. I should share the screen. It will be much more clear. Okay. Here. So what we are going to see is the timeline here. The first block is present of one stimuli, and the second block is present of the second. And then the mouse should leak or don't leak at this like reddish blocks. And you can see here at the match trial, which means that two stimuli are the same, they should leak to this center port, which you can see uh, the block is covered by this black line, but in the non-match trial, they shouldn't leak. So you can see they never leak. At here, it is almost blank. So the probability of leaking will then transform to a distance. The longer the distance, it means that the mouse perceive it as more dissimilar. Yeah, oh, okay. Share screen again. I should explain the distance metric. The metric here. Okay. DAB. For example, if A and B are two stimuli, if let's assume A and B are very similar order, and 1 minus probability of no-go of A and A definitely the same. So it will be 0. 1 minus 0 equals 0. 1 minus also same stimuli. 0 equal 0. And A, B, they are very similar. Presumably, they are identical. It is also 1 here. So 1 minus 1 give a distance of 0, which means A and B, they are overlap in a perceptual space. Okay. Then they show the similarity of order and using MDS to visualize the distance between multiple stimuli. And sorry to share it again. Here, let's see the dissimilarity matrix. On the X and the Y axis, each color indicate one stimuli or one kind of smell. Let's only focus on this lower triangle. The higher the distance, the lighter the color. So which means that the mice perceive this smell as very dissimilar. And then researchers combine some smell together. As here you can see the orange and the sky blue, they combine together. So presumably these two should have because they are overlap with this orange, so they should be a little bit much more similar to the other block, which you can see there's a darker color, which means they are much more similar in mouse perception space. And what I found interesting is after they converted to distance, you can see here and here, the orange and the sky blue corresponding to this orange and this sky blue here and here. So, oh shit. Oh, I cannot delete. Okay. So this means that the combination of smell seems much more similar to individual component of this combination. So this is very interesting, and that this kind of this kind of result actually supporting that their paradigms actually measuring the perceptual similarity of the smell of this smell. Okay. Yeah, let me stop sharing. And finally, they ask one more question. They are asking, do different mice rank this order pair in a similar order? In other words, is there any significant individual difference? Which means that some mouse can have a different strategy to do this similarity judgment, and this is very different to the other mice. So they first rank like all 276 order pairs from shortest to the longest distance based on average over all mice. Okay, it is in figure 3C. Then they group this ranked pair into bin of different size. For example, the top four as a bin, and the fifth, the sixth, seventh, and eighth as a second bin. So they group this rank pair into bin of different size, 4A, 16, 32, or 64. For each bin, they calculate the average distance for those order pair, and they do this for both individual mice and across all mice. And what I found most interesting is that they found as bin size increase, the correlation approaching 1.0, which means that the mice increasingly agree about the relative ordering. So with this largest bin, which is 64 pairs, all mice perfectly agree about which quantile each order pair belongs to. So yeah, this shows that while mice might disagree about this kind of fine-graining distance differences between specific order pair, but overall they show very nice consistency in their border perceptual organization of this kind of order pair. Yeah. So the perpetual map is quite similar across individual mice. So to sum up, this is a paradigms or method paper that's showing that they develop a reliable and robust paradigms to detect the perceptual similarity between stimuli in mice model, and in future maybe we can change the stimuli to from order to like visual stimuli or some kind of sound stimuli. So this paradigm actually, so it's versatile. Okay, that's all from me. Thank you.
Any questions, comments so far?
I think I skipped some content, but do you want to add them?
If no one will, I do have some question regarding the bin size and the related calculation. So at 64 bin size, the correlation, as you said, is 1. But I don't get how it could possibly be 1 for all 10 rats. If you look at the 3C, the graph 3C, so there are 276 older pairs. If we bin them by 64, there's like 4 dots that should be there on this figure. So 3C is a 32 bin size figure. If we're imagining a 64 bin size, instead of having 9 data points, we'll only have 4 data points. But some of these, like one of these rats are almost consistently rating order to be more distant from each other.
Which one?
So the X, the one marked with a little X. Should I share my screen if it's difficult to see? Okay. So what I mean is we see that the X represent by X. This rat is consistently rating distance higher than other rats. So if we bin these two data points into one, these two data points into one, so on and so forth, it's difficult to imagine the Spearman correlation would be essentially perfectly one between all of these rats, right? Or am I misunderstanding Spearman correlation in some way?
This figure show the being of 32, I think.
Yeah.
And the Spearman correlation is about the rank, and this X is about the rank. So the first X should correspond to the. They don't care about the value also. Maybe let's find another similar example, which is one of the triangle, maybe.
Yeah, sure. Let's see. Triangle.
A triangle, but but one vertex pointing to the right side, which is upper. The value should be 1.
Oh, I see what you. Oh, okay.
Yes, this one.
Right.
And then what we are looking for is the first symbol at the 32 should be lower than the second symbol at the 64, and the 64 should lower than the following one. So this is the rank. If they are if they follow this kind of step for one mouse, and the other mouse, which is the triangle that also located at 1, they also have this kind of rank, then it will be perfectly matched because the order is 1, 2, 3, 4, 5. But if the triangle at the maybe other
sorry, just quickly. When we're talking about rank, we are talking about like for one mouse between the like within its own data points. Is that the ranking we're talking about? Are we talking about comparison between the different mouses?
ranking for one mouse. Oh, and then they are comparing between the comparator is between one mouse and the average of all mouse,
Right,
which is
I
the
see.
black line there.
Okay.
That's my understanding of Spearman correlation and here and the being no and the how to calculate it here.
Oh, I see.
Ah.
But just to like, I think when all the mouse have the Spearman correlation of 1, what does it mean? What does it suggest about the data exactly?
I think my interpretation would be, my tentative interpretation would be that all mice, well, for a particular mice, its response pattern or its sort of distance measure is highly correlated with the group. So therefore, you could say that. So therefore, you can do that for every single mice, and then therefore you would say every single mice responds similarly to as other mice do.
Okay.
I think that is what they are trying to say here.
Yeah, they are comparing each individual mouse with the average of all the mice.
Okay, maybe
in a ranking way, right? Yeah.
Okay. I think I understand better now.
But there is still the problem of the diamond mouse, which I don't quite understand. Mike, do you still want to share screen, or do you want me to?
Yes.
Because
I
it will
can
be on
share
the same figure.
Yeah. No.
Yeah, if you look at the diamond one, that would be, if I could highlight it. Where is? I don't have annotation. Hold on.
Yeah, the bottom one. Yeah.
You have the at the end 276 there is the triangle one, and then beneath that there is the little diamond one. And then you can see the diamond one, if you trace it back, yes, that's the one. And then it traces back, it actually goes down. And I don't understand what that means in terms of the analysis. Yeah, like could you still get a correlation of 1 there?
Oh wait,
If
diamond is not a correlation of 1. You can see it is almost the lowest, 0.8.
yes, but at the end 64, they say every single mice has a correlation of 1, right?
Yeah, I see. How can I know?
It's the bin size is too large, I will say. If the 64 is just
4
divided
points.
into 4 or so.
34. So I think 30. There's also diamond here, right?
Yeah, that's right.
This one is 64. And then this one and this. And then if we take average, this two will be here. And take average, this will be here. And this diamond, and where's another diamond?
So these two at the end, right?
So I think that's the problem.
Yeah.
Right?
Oh, but they discard the final bin, right?
Oh, right.
They
So
discard final bin. Okay.
It's 2, 2, 2, 2. So the last one doesn't matter, and everything makes sense.
Yeah.
Okay. Yeah.
Okay. Yeah. Then that makes sense. Okay.
So if I say if I give analogous, it will be some kind of GWOT, but in K larger than 5, this kind of cluster alignment. if we just give a rough explanation.
And make the tolerance really high.
Yeah. For example, this is red, pure red, and a little pink, and some reddish color. They are ranked like that. And then this greenish color. So if we bin them together, that the conclusion is, the red is different to green. So the larger the bin, the larger the cluster, or more abstract the stimulus, the property of stimulus. I would say that. Yeah.
Let's see. I've got a comment, which I don't know because I'm still learning about different types of dissimilarity rating paradigm and how it's different from discrimination task. One thing that struck me is that what they're doing is to use the probability of no-go as a measurement of distance, right? Or use that to calculate distance.
Yeah.
And the emphasis I wanna that sort of struck me is on the probability of no-go choice. And therefore, what they are testing essentially is the consistency of saying different, or the consistency of judging different, as opposed to measuring the magnitude of the difference. Does that make sense? Or in other words, what they're doing here is to say that if I have two colors, two odors, and the mice consistently say that they're different, then they are very different in magnitude. And I'm not sure if this conflation is common, and is it justifiable to say that the consistency of the difference is equal to the difference, or the magnitude of the difference. Because I can imagine saying color, when we perceive color as an example, we may say that orange and red can be consistently different, and then red and green can also be consistently different. But then we could also say that green and red are more different than red and orange, if you know what I mean. Does it make sense to you what I'm saying?
I think
Yeah.
I get what you mean, and
Yeah.
it's probably hard to answer from this paradigm. I think if I remember correctly, we can trace back to the origin of such paradigm. I think originally like in 60 years ago, it was called stimulus generalization. It's not so say if we don't put two stimuli there, we just put one, say a color, a green color, and we have it's a continuous scale of color, say gradually change from red, orange, yellow, green, and then blue, and then purple, yeah, something like that. And if we only reward the animal for green color, and then but we present them a continuous I mean for example 10 or 15 continuously changed color patches, and you will see a gradient the frequency the animal respond to the color gradually decrease as the distance get further from green.
So it's sort of like a symmetrical Gaussian,
Uh,
or maybe not Gaussian, but it
yeah,
is
not Gaussian.
symmetrical.
You
Yeah.
will see
Yeah.
you will see the consistency and
Mm.
continuously change.
Ah,
And I mean, as the physical distance between colors increases. I think that's the rational or theoretical, sorry, evidence basis of such a paradigm. That's what I
I see.
understand.
I see. I see.
Yeah.
Ah, is that used in animals, or has it been? I assume it will be used to justify this type of perceptual distance measure in animals. At least probably they would include something like this.
From my
Okay.
knowledge, I learned this paradigm from pigeon studies. Yeah.
Ah, okay. I see. I see. Mm.
Because
In
for
terms of
humans,
the
it would be too easy. Yeah. Okay.
In terms of the, I also want to add some something more because similarity at last century, it is kind of principle of association and also what Chuyin say generalizability and transfer, stimulus transfer, like looking at the one stimulus and then we know it is similar to the others, then because pigeon or the animals think they are kind of similar. So the behavior that responding to the sample or the reference stimuli will
Yeah.
also generalize to another they think similar the similar that they think the stimuli that they think similar. Yeah.
Yeah, I think I know what you mean. Yeah,
Yeah.
Yes.
I see.
Yeah. So, or it might be understood as probability of confusion, something like that. Yeah.
Mm.
Uh, yeah, so I think using probability is a good way to infer this kind of distance.
Mm.
Mm.
At least at the start, right? I think one way in which they could sort of mitigate this, or at least trying to go around this, is to use the side panel. So they have the central faucet or whatever that they use to dispense the water, and then they have a side one as well, right? And what they're doing is to only measure responses to the center to say that if you lick to the center, then that means you indicate they're the same. And then if you don't lick at the center, then that means that they're different. And one way to sort of go around this type of probability, well, actually, it's still probability, but it would be where you can incorporate also the side faucet to say that you use the side faucet to indicate that this is dissimilar and the center as similar. So you're measuring, so therefore, I don't know. I feel like that could be I don't know. That feels to me a more straightforward paradigm than a traditional go no go. Although how much it is related to probability now, I verbalize it. I realize that it probably is a different direction than I intended. Yeah.
Sorry, I didn't really get
So
it.
okay,
Are you
so
saying?
the idea is that it would it be better to include the side faucet as a response location. So
Okay,
at the
so
moment, the
to
center is the only location that you will measure response, right? If
yeah,
you
yeah,
lick center, that means same. You don't lick center, that means different. What I'm saying is, could we also add a side faucet measurement to say that now you train the animal so that center means similar, side means no similar, oh sorry, dissimilar or different. And then no response just means you're not paying attention to task or some other problems that they just make you not respond basically, or you're not sure or something like that.
I will say
And
this will increase the difficulty of training because
Yes.
they only have two options and one of them will guarantee the reward. So if this one is, say if the center is wrong, then they leak the peripheral region or so, or the side region, and then they get rewards. They don't know. I mean, it's too easy to receive the rewards, and it's basically guaranteed that.
wait, I think in both conditions, leak or in both situations, they got a reward. They are very thirsty mice. And the difference is if stimuli are the same, they can get reward by leaking the middle port. But the reward is not instantly get it by leaking, but by their behavior. So when they do correct, so-called correct behavior, they will eventually get a reward. So animal want to get a reward because they are really thirsty. I think in the supplementary material, the experiment actually started before 1 week before, so they are already water deprived before the experiment start for 1 week. So yeah, if you want to test the attention, I think uh, uh, yeah, the water deprivation already forced this happen.
I see.
Yeah.
Yeah, you're right. Yeah, attention wise, yes.
Mm.
Mm.
I have a question regarding this paradigm. So this experiment present stimulus in a sequential way, and I want to kind of justify this sequentially present stimuli, but I don't know whether it is correct because I want to justify based on IIT axiom, which is exclusion, saying that experience is definite and it is whole. So every time, single time, we can only experience one feeling. So sequentially presented stimulus is kind of can be justified. I don't know. Do you have any
I mean,
comment?
I feel
there's
like
a practicality issue,
mm,
right? I mean, how if you present, how would you do if you have the same odor?
Mm,
You present it twice and ask them for similarity?
mm,
I mean, I don't think that makes like I don't think there is a really good way to do a simultaneous presentation of two odor. Maybe there is a reasonable way to do it if you have visual stimuli or sound stimuli. But odor seems very difficult to conduct simultaneous presentation.
yeah. I'm
but
not sure about mice, at least for humans. Like we've got two nostrils. Technically, we could do, but the idea will be like left
Oh,
nostril one stimulus and then right nostril another stimulus. But the problem is that I think the nostrils are connected before going to the olfactory bulb, where so there will be a problem of you're actually just doing a mixture rather than comparison of two, I think. I'm not sure a similar thing could be said about mice. They probably have a whole different apparatus to smell, which I'm not familiar with.
mm,
Uh, but yeah. Yeah, I thought
right.
Mike, you're gonna say something about the Sino. Sorry, not the Sino, like the presentation order effect, where like
I mean,
presenting one like just affects how you perceive the other, something like that.
they essentially showed what one would expect, right? They
Mm,
have what 17% of is a triangle inequality.
Yeah.
Uh,
mm,
Odor, I'd say the asymmetry condition was violated. So they are expecting 5% of chance level violation. They actually got 17%, which seems to be, I mean, it's a good indicator that for some color pair, I mean, older pairs, they are asymmetrical. I mean, we can talk quite a bit about the nature of perceptual distance and what's the issue with it, and why perhaps vector space provide a better solution, but it's not central to really the what this paper is trying to do, right? It's just trying to develop a good perceptual distance methodology in rats, in mice, and from my perspective, they have done it pretty well. That's my impression, at least.
Yeah. Yeah. Okay. And in terms of the similarity between odor, I remember in some kind of a fragrance shop, they will give us a coffee bean when we smell a fragrance, and then we want to refresh our smell. We smell the coffee bean and then take another fragrance. I don't know, can it ameliorate this kind of odor effect because it actually refresh the feeling of smelling. Yeah.
I think rather than refreshing, it is reestablishing a baseline. But the baseline itself, yeah, so using coffee, this coffee itself is quite a strong smell. That's why I think they're using it as a refresher, so to speak. Because then you can quickly return to the coffee baseline. Then you can say coffee, then smell A, then go back to coffee quickly, then to smell B, and then you can have this. But then you will still have a baseline stimulus interaction problem. I think this is a bit unavoidable, in my opinion.
Yeah, I think what you said, it really makes sense. It is a distance toward a coffee bean. Yeah. Okay.
Just like what's the least requirement for similarity, any similarity to make sense? And as far as I can tell, the only real requirement is that if you present the participant with two of the same thing, be it same color, same contrast, same smell, same sound, if you present two of the same thing, they should say, yes, they are perfectly similar. That seems to be the only requirement for any similarity measures to make the basic amount of sense.
Really? Is that consistent with quantum prediction?
No, this is just a personal thought. Because
Okay.
you imagine the participant, right? So you present them with color A, and then you present the other color to be A, B, C, D, E, F, G, whatever it is.
Yes.
And every time you present them with a color pair, they say, yes, they are perfectly similar. They give 8 out of 8 rating or whatever. Like if they are answering in good faith, I mean, can we just say, oh, you don't have any sensitivity in your color experience, so your answer are invalid or inaccurate in some way? We can't necessarily say that, right? Maybe they just only see one color. It doesn't make a lot of sense, but it's not, I guess, not impossible. But the only thing that doesn't make sense is that when you present color A versus color A, and they say, nah, they are completely different. That's the only circumstances where everything breaks down fundamentally. Does that make sense?
Yes. If they say completely different, then it doesn't make sense. But I wouldn't say every time they will say that's exactly the same.
But maybe they like in terms of are we considering noise, or are we considering genuine perceptual difference between the same stimuli?
Genuine perceptual difference under some circumstances, like not different, like specific background color, like presentation durations, or some other random situation of the participants. Yeah.
At the very least, the same stimulus would differ, well, depending on whether you want to do a simultaneous presentation. In this case, that will be differ by location.
Yeah.
Or if you want to
And
do
also,
if you do a sequential, then there will be differ by the temporal dimension,
yes.
where one is after. So it is still, you could argue it is still the same stimulus, but there are dimensions to which that they are different.
Yeah, that's right. They are just remind me of category theory. Oh, they're different kind of the same, have different levels. And it's not perfectly equivalent, even though they are technically the same color.
Yeah, right. That's what Matafumi said last time. Okay.
Yeah, that's me. I don't have any not much else to say.
Yeah, I have a like comment or something. I think the interesting part is they compare an older structure with some previous studies on humans based on the chemical features of their stimuli, right? And it might not be feasible in smiling, but might be feasible in visual stimuli like colors. Based on the structure of a visual system, we can predict a similarity structure from it, like we have three types of cones, and each of them activate differently to different wavelengths of light. Then we can predict for specific colors, specific pair of colors. We should make a specific judgment of similarity. Yeah, but actually the subjective feelings will be different. That's just some random thoughts, and yeah, from me. Yeah, we can do it. I think it's more feasible to do it, or it's only feasible to do it on animals, not humans, for some other kind of experiences. You can cut their cortex and make some predictions based on the structure of it.
Oh,
Yeah.
we don't need to cut them. We just need to inhibit it using the optogenetic technique.
Okay, yeah.
Yeah, using light to inhibit. Yeah, that's feasible.
It's kind of the main benefit of this study, right? The
Yeah,
idea
yeah,
that
right. It, mm, yeah, so currently we only have physical distance based on the physical models like art in terms of colors, sorry, because I'm only familiar with that, based on RGB values or CIE lab models, and that's a physical thing. And we have a subjective space from our subjective ratings. But I think there might be something in between, like the similarity structure predicted from our biological structures. If everything, it's a bit hard to explain, or say it's just based on our biological structures, the activity of our biological structures, we might get a similarity structure, and which might be different or similar to our real subjective experiences. Yeah.
right. So like similarity between neuroactivity, that kind of thing.
I don't want to limit it to neural activities. Yeah.
I see.
It would be difficult. I mean, I mean, like for color, I think a color perceptual structure or a color space is a very well, it's very common. But if you think
Yeah.
about older, like
Yeah.
or maybe even in what I'm doing in sound, like the sound structure, it's actually difficult to graph it out. Like we don't have a lot of descriptors about it, and it is very finicky in my opinion. Like these things are these structures are
Yeah.
difficult
Or
to characterize out.
it's very complicated, I would say, for
Mm,
sounds, for example, the structure of our ear and audio system.
mm,
I think we have not completely understood it, right?
mm,
That's
yeah.
definitely a difficulty, yeah.
Mm.
Just to make sure I understand you, do you say talking about like phenomenal structure, which is qualia structure, should mirror in our neural structure kind of thing?
Potentially, yeah, that's an integral question. If it's exactly the same, then we can say that.
Yeah.
Okay, I see. Not really exactly the same, but they are existing a very nice transformation that can generalize to all stimulus. Just one transformation, and it's consistent across all stimulus. Then transport phenomenal structure to neural structure. I think that's also possible. Like transportation, only one algorithm. Transportation plan,
Mm.
like folding, or the way of
Yeah,
folding.
yeah, the structure gradually changes,
Yeah,
something
yeah,
like that.
yes.
Yeah, as the processing goes deeply. If I mean deeper and deeper, yeah. Mm. Okay, yeah, that's some random thoughts from me, and
Not
yeah.
random thought. It is actually from the paper of structural constraint of neural, a structural constraint of neural correlates of consciousness. I remember that paper also proposed same
Oh,
thing.
well, yeah, in terms of neural activities, yes, but I kind of now I think it might be wrong. Initially I kind of wanted to extend to some other physical structures,
Oh,
like mm, yeah, maybe cones.
hmm,
Cones are a type of neurons. I don't know,
hmm, concells.
Oh,
or well,
so the state of the measurement device is essentially what we are talking about.
yes, it's detector, like detector. Yeah.
Yeah.
Oh,
Or it might technically it is a part of neural activities. Yeah. So yeah. Anyway, this point I might be wrong. Yeah. Mm.
photoreceptor.
So what you want is like a functional bridge between the external, the structure in the natural world, the external physical world, and the subjective world. And based on the description, yeah, it seems functional, right? Is the primary way that you have carried.
Mike, I think you dropped out in the last bit.
Sorry.
Yeah.
Oh, sorry. Yes. Yeah. Sorry. There's possible connection here. So it is. It sounds like a functional process of information being transformed from the external world, how it's processed in the detector, the eyes, and then the processors, the brains, and then somehow
Come to experience.
to experience. So that's
Yes.
what you are. Okay.
Yeah. Different levels. Yes.
Yeah.
Mm, I see. Okay. Okay. Yeah. Anyway, never mind. It's just leave it. Yeah.
Okay.
Mm, okay.
Um,
Mm.
If I may, I've got a final question. Probably coming back to the probability consistency versus magnitude I was talking about earlier. I'm wondering if response time could be a measure that they take into account as well, where quicker responses to well, actually, no, it will be difficult because then you can't say that they're quicker at not responding, which makes no sense. Actually, now I think about it, it's actually difficult. Um, yeah, don't worry, don't worry. I had a thought and then I thought otherwise.
Okay,
Because what I was thinking was to use response time as a measure of magnitude, where quicker the response, the bigger the magnitude of difference they indicate. As a behavioral measure of magnitude. But then yeah, as I said,
I
it
think
will be yeah.
if you have two buttons, something like two buttons,
Mm,
and you need to press one of them, then I think reaction time could be a measurement.
yeah,
But here it's a no, it's a go no go paradigm.
yeah. You can't be quicker at not doing anything.
Yeah.
Mm.
And also in the figure 1C, you can see even in the during the presentation of order, they are still leaking.
True.
Yeah, so
Yes.
they have a baseline of just start licking the thing.
Mm. Mm. Yeah.
Great. Okay. Any more comment or question? Nope. All right. Thank you very much. This is the
Thank
conclusion
you,
of this
thank you.
paper. Thank you.
See you next time. Bye bye.